So I'm Sean Eddy. I'm currently a professor at Harvard University and a Howard Hughes Medical Institute investigator. And I was involved with Rfam from the start. So I was a postdoc at the LMB in Cambridge. The computational biology group was part split between the LMB and the then brand new Sanger Center, before it was the Sanger Institute. And we had developed the PFAM database as part of my postdoc with Richard Durbin and Eric Sonhammer. Alex Bateman came on, and there was a lot of interest in doing the same thing we were doing with PFAM for RNAs. And so that was the birth of Rfam when Sam Griffith-Jones came along and I think was looking for projects. So the initial vision was to do just what we were doing with PFAM, but for RNAs, for structural RNAs, and to be able to take a new genome sequence and annotate it for all of its RNA genes. We were fresh off the Human Genome Project, which we published in, 2000, and doing the annotation of the RNAs in the human genome was something that I and someone in my lab, Tom Jones, had done pretty much by hand. And we really realized the need for some sort of genome-wide comprehensive collection of the sort of standard set of non-coding RNAs, especially in eukaryotic, well, and really in any genome. And so that's what was sort of sitting there waiting as a project when Sam Griffith-Jones came along. Well, there's a whole bunch of them. And I guess I should first off say that I didn't really do much for Rfam proper. That was really Sam and Alex and other people. My lab quickly became like the underlying foundation for both PFAM and Rfam, but all the real work for curation of the families and stuff rested at the Sanger and the EBI. So we provided the software, and so a lot of the challenges that we faced were really the underlying software issues, and a lot of the curation issues were things that Sam and Alex had to face. So on the software, the main challenge was really the compute intensity. Sam had to set up a sort of hinky pipeline of using BLAST as a filter before you presented anything to the SCFG software. And that was totally offensive from my point of view, because the whole intent of developing the SCFG software was to have more powerful searches than BLAST. So even though, you detuned BLAST so that it would, have a lot of false positives and you let those through to the SCFG stuff to filter out, it was still pretty offensive not to be able to run Infernal at genome scale. And so that became a focus of a lot of my lab's work for many years, was to find ways of accelerating Infernal. And when Eric Naraki joined my lab and really took that on as a project, he and Diana Colby, two graduate students in the lab, put a lot of their graduate student effort, and then Eric later as a software engineer, now at NCBI and still part of the project, they really did a ton of work to get Infernal to the point where we can use it like at all at genome scale. So now Infernal is about as fast as Hammer used to be before Hammer itself was sped up by related techniques. So I would say that's the main thing from our perspective, is just the compute intensity of the SCFG software. We needed to do a lot of work on that to make Rfam practical. One thing that I did not expect, that I'm really pleased by, is how much the computational biology community has used Rfam. So I really, coming from human genome annotation, I had really seen the need as an annotation tool for biology. But what I hadn't appreciated and didn't see coming was how much the RNA computational community needed good benchmarking and testing tools. And so to have this comprehensive set of a couple thousand different RNA structures with alignments as a test bed for new structure prediction, new structure finding, whatever, that's been a really pleasant surprise. And so we've got the Banasque, are you going to Banasque? We've got the Banasque meeting coming up in August here in Spain. And that whole community has been meeting like every 3 years for forever, since the beginning of Rfam. And to see how many computational RNA biologists are using Rfam, like every talk will be like, so we developed this fancy new method and we took Rfam and benchmarked it. And that's just wonderful to see. That's a use of the resource that was an extra surprise. That's a great question, because my ability to predict the future always turns out to be wrong. I think that we'll have to see. I think that what we know is that it has become this central resource for both annotation and for computational method development worldwide. And so, we're going to see more of the same. We keep collecting sequences. We keep discovering new structural RNAs. We're going to need new models, so the Rfam is going to grow. We're going to get better at solving some of the annotation problems that we face, like telling the difference between pseudogenes and real genes that are under structural constraint. So that'll get stronger and stronger. And I don't know, RNA biology keeps throwing us curveballs and interesting new biological surprises. And so, Rfam's ability to sort of keep up with RNA biology growing is going to be interesting to see.